So good morning, students. This is our third video on Chapter 16. It's kind of the wrap-up material toward the end of Chapter 16 on DNA structure and replication. So let's see where we're headed with Chapter 16 material here. I hope things are going well for you and that you're able to keep up with these short videos. So we're here. We've looked at how DNA, what its structure is, and how it's replicated. But there are a couple other wrap-up topics in Chapter 16, and one of them is about what happens if DNA is damaged or if replication isn't perfect. How do our cells prevent mutations from happening? Because mutations in DNA are really bad. They are going to be passed along from generation to generation to generation, right? Our cells want to make, when they replicate their DNA, want to make a perfect replication. But mistakes are going to happen, and DNA is going to be damaged because of exposure to chemicals or exposure to environment. DNA can be damaged in a lot of different ways. So what kinds of mechanisms do we have in place to repair and replace damaged DNA and to prevent mutations from happening in the first place during replication? A lot. It turns out that humans have over 130 different DNA repair enzymes that do the job of helping to deal with messed up DNA. The first thing that is worth mentioning is proofreading. DNA polymerases, as they build new DNA, proofread the DNA. If they make a mistake and put in the wrong nucleotide, they typically go back, take it out, and put in the correct nucleotide. It's kind of like if you were typing and you know you made a mistake, you automatically hit that backspace button and retype the right thing. And it's just kind of muscle memory almost. Well, DNA polymerases proofread as well, right? So here's a picture of a double-stranded DNA molecule, and in this double-stranded DNA molecule, it looks like we're building this yellow or this white blob here is a DNA polymerase building new DNA. It sees a G, it should put in a C, but oops, it puts in a T initially. But then polymerase detects the mistake. It realizes it, it pulls that T out and builds in a new nucleotide. It puts in a C, not shown on the screen. So proofreading is really important to prevent mutations from happening during DNA replication. But sometimes mistakes aren't caught, and sometimes DNA gets damaged. And so there are repair mechanisms for dealing with problems in the DNA. One of them is called mismatch repair. Another is called nucleotide excision repair. Basically, mismatch repair is dealing with a small problem, a single mismatch between a pair of bases. Nucleotide excision repair deals with a bigger problem, a bigger mutation. And so we'll show you an example of mismatch repair first. Okay, so we can kind of look down here and look at mismatch repair. Okay, so here's a picture of a molecule of DNA that has, it's been newly synthesized, so we see that there's a template and a new strand of DNA, and there's a mistake. There's a G that's matched up with a T. Now, what mismatch repair enzymes do is they recognize that mistake. Oh, there's a mistake here. G can't be paired with T. That's a disaster. It is a disaster. And what they'll do is these mismatch repair enzymes will actually remove a chunk of DNA right by the mismatch, and then a DNA polymerase will come along and build the new DNA again. And then ligase will seal the gap, and we'll have a wonderful repair. Now, the question I have for you is, how do the DNA repair enzymes, after they see a mistake, oh, yep, I see a mistake, G is not supposed to be paired with T, how do they know which nucleotide is wrong? Is it the G that needs to be replaced, or is it the T? Well, you say that's simple. They just have to know which strand is the newly synthesized DNA. Presumably the new stuff is the bad stuff, right? Okay, how do DNA enzymes recognize the newly synthesized strand versus the template or old strand of DNA? They do. They recognize the old strand versus the new strand of DNA. They replace and repair the new DNA. They assume that the new strand is wrong. But how do they tell the difference? Well, it turns out that after DNA is made, methyl groups are added. Methyl groups are like CH3. Methyl groups are added to DNA molecules. The template strand will have more methyl groups attached to it. We'll say that the template strand is more highly methylated than the new strand, and a DNA repair enzyme will see that and go, okay, now I know the new strand. Now I know that it's the G that I need to remove, and I need to replace that with something that matches T. I need to replace it with an A. But it can recognize the newly synthesized versus the old or template strand of DNA because of methylation. Pretty cool, huh? Nucleotide excision repair is just another mechanism to repair a chunk of damaged DNA. Usually the damage is bigger, so you have to take out a bigger chunk of your DNA. For example, I just wanted to give you this example. Thymine dimers are fairly problematic in skin cells because ultraviolet radiation causes two thymines that are next to each other, so they're paired up with adenines, can cause two thymines to kind of pair up with each other in kind of a buckling sort of fashion. They make the DNA kind of buckle up, and that doesn't look good, and it can't be replicated properly. DNA polymerase, when it sees it, is like, I don't know what to do with that dimer sitting there. And so DNA repair mechanisms see and find the dimer, take it out with a nuclease that takes out DNA, replaces the DNA polymerase, then goes in and replaces the DNA with new DNA that is appropriate. The dimer has been removed, and then finally DNA ligase comes along and glues together the pieces of the newly synthesized with the old DNA. Whew. Thankfully, we have these kinds of repair mechanisms so that we can prevent mutations as our cells are replicating. Right? Remember, things like cancer cells have kind of uncontrolled cell division in part because they have so many mutations in their genes that regulate cell division. Mutations in their genes kind of mess with the regulation of cell division. The more mutations you have, the more likely you are to have problems, and then you get a cell that is really out of control. So having good repair mechanisms in place is critical for managing cell division. Cancer cells often have problems with their repair mechanisms. Okay, one more topic here, our topic number 2 of this video, are telomeres. Telomeres are the ends of the chromosomes, and we're going to take a look at this information here on the next slide, and then we'll ask these questions as well. So when we're looking at a chromosome, it turns out that at the ends of the chromosome are a set of DNA in humans, TTAGGG, that is repeated over and over and over and over again, like 100 to 1,000 times in humans. So we see TTAGGG, 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 etc. over and over and over again. These are not genes. The ends of chromosomes, the telomeres, are not genes. They don't code for the building of proteins. They're just there, sitting at the end, a whole bunch of repeated nucleotide segments. People are like, why do they exist? Well, we think they're protective. They're protective caps on the chromosomes. The chromosomes are filled with important genes. The telomeres kind of protect the chromosome. And let's take a look at how that happens. Let's just make sure that I kind of got through everything except this. Okay. It turns out that when you're replicating DNA all the way to the ends of a chromosome is really hard to do. And so what happens during replication, and this is true, and you have to believe me, and I'll show you a picture, but as you replicate a whole chromosome, the ends of the chromosome with each round of replication get a little shorter. And so a cell that's replicated over and over and over again will have shorter and shorter and shorter chromosomes. And what gets shorter is the length of the telomere. The length of the telomere gets shorter and shorter and shorter with each round of replication. It's good that the telomeres are out there at the ends of the chromosomes and not some important genes of yours, because if you had important genes out there, they would be getting cut off and getting shorter and shorter and shorter, and pretty soon, obviously, the genes won't work if they don't have all the genetic material. So let me try to sort out why replicating the ends of a chromosome is problematic. Why do telomeres get shorter? Okay. This figure tries to point you to it. So now we're looking at the very top component here. What is happening is we're replicating a molecule of DNA. The dark blue lines represent the original template strands of the molecule that have separated so that we can replicate it. The kind of light gray lines here represent the newly synthesized DNA. And what we've got is we're at the end of a chromosome. The end of the chromosome on one side is the leading strand of DNA that's being replicated continuously all the way to the end of the chromosome. No problems there. But on the lagging strand, remember that we had to add a primer and then a primer and build. So here's what's happening. You add a primer, you build new DNA. You add a primer, you build new DNA. You add a primer, you build new DNA. A primer, you build new DNA, etc. Okay? Oops. The red component on this drawing are the primers. Oh yeah, you can see that kind of in big over here, right? So you put in a primer, you build new DNA, and then you have to replace the primer, right? DNA polymerase replaces the primer, and it's all good, except the primer at the very end of the chromosome. The DNA polymerase can remove it, but remember, DNA polymerase is kind of quirky. It can only build DNA when there's something to add on to. And so here, DNA polymerase is like, I don't see anything to add on to. I can't build anything here because there's nothing for me to attach my nucleotides to. And so with every single round of DNA replication, the chromosome actually gets shorter. It loses a piece of DNA every single time the cell replicates its DNA. The chromosome gets shorter. Whoa, that's funky. It really happens. So our telomeres get shorter with every round of replication. And let's think about this then. If you look at the telomeres in my cells and compare them to the telomeres in your cells, my telomeres should be shorter than yours in all of my cells because they've undergone probably more rounds of replication than your cells, right? That actually happens. The telomeres in your grandparents' cells are going to be the shortest yet. They're going to be shorter than your parents and shorter than yours. So in aging cells, telomere length is shortening. But wait a minute here. You can't have telomere shortening forever because then when you make your gametes, when you make eggs and sperm, are they going to have shorter telomeres with each generation? And then pretty soon we're going to have, no chromosomes left over generation after generation. No, that doesn't happen. In germ cells, those are cells that are going to give rise to sperm and to eggs. The telomeres are lengthened back to what's considered normal conditions by an enzyme called telomerase. Telomerase is an enzyme that's active in germ cells that rebuilds the telomere and makes it longer again. That adds more repeats to the ends of the telomeres. And that happens in all of our germ cells so that at least our eggs and our sperms are having long telomeres in them. But this enzyme is not active in our normal body cells. Now this is really interesting. cancer cells are cells that just divide and divide and divide, right? And they keep on dividing. Sometimes we even say they're immortal because they just keep on dividing. How do they deal with shortening telomeres? Because eventually, wouldn't they be unable to divide if their telomeres got so short that they disappeared, and then you had, cutting into the chromosome? Wouldn't cancer cells just die? Turns out in cancer cells, that enzyme called telomerase is active. So cancer cells have reactivated their telomerase enzymes so that they can lengthen up their telomeres as well. So in cancer cells, the telomeres are often quite long. And researchers are really interested in this because if you could somehow target that telomerase enzyme and prevent that from working in cancer cells, that might lead to the death of the cancer cell over time. Who knows? It's a really exciting area of research. Okay, you have to study this slide for a while. Last topic, packing of chromatin. And so we've been talking about DNA, and we've also talked about chromosomes when we talked about cell division, right? We said, all right, during cell division like mitosis or meiosis, the genetic material, the chromosomes, coil up. The DNA coils up on itself and forms these chromosomes. How does that happen? How is DNA actually organized? This is a list of a few terms that you should know. It just helps us understand how the DNA is packaged inside our nucleus and how it's managed. Here's a picture of it. So here's our double-stranded DNA molecule, right? It's very, very, very small, 2 nanometers in diameter. How is it packaged? How is it organized? Well, all DNA is associated in eukaryotes with what are called histone proteins. Those are the purple little things with tails there. The histone proteins get wrapped, sets of histone proteins are wrapped in the DNA. And then you have what's called a nucleosome, which is essentially a chain of DNA with histone proteins. That's 10 nanometers in diameter, and the nucleosomes look a little bit like beads on a string, look like a little necklace. And so that's what language we use. And then the DNA gets more tightly packaged into 30 nanometer fibers. So these histones and these the DNA get kind of wound up into bigger fibers. All of this material we call euchromatin or true chromatin, because the way that DNA is packaged as a nucleosome or as a 30 nanometer fiber allows for DNA to be transcribed and translated into proteins. There's some enzymes and machinery needed to find the genes, to transcribe the genes, make RNA copies of the genes, and then those RNA copies can be used to make proteins. If the DNA is packaged really tightly, then genes can't be transcribed or translated. But the DNA that's packaged in these ways is called euchromatin because it's loosely packaged. It can be used to make proteins. Then, however, the 30 nanometer fibers can be made into 300 meter looped domains. It's sort of like there's a scaffolding that loops these fibers up, and so you get this kind of tightly packed 30 nanometer thick fibers, 300 nanometer thick fibers. At this point, the genetic material is called heterochromatin, other chromatin. It can't be used when it's packaged this tightly. It can't be expressed. We can't get in there with machinery to make RNA, and then the RNA is used to make proteins, right? Because it's too tightly packaged. And then when we're moving chromosomes around during mitosis or meiosis, we have our DNA packaged really tightly into what are called metaphase chromosomes, or 14 nanometer squished up genetic material. That is very tightly packaged. It's much easier to move around than the loosely packed DNA or nucleosomes, right? And that's how our DNA is moved around during mitosis and meiosis. But it's tightly packed. All right. So we can package DNA in different ways depending on what our cell needs. If our cell needs to do mitosis or meiosis, pack it all up really tightly. If our cell needs to be using genes and transcribing them and translating them to make our proteins, we have to have more loosely packaged DNA. That's it. Chapter 16 has some test your understanding questions. I strongly recommend looking for animations of DNA replication too for Chapter 16. Animations are going to help you understand replication in a way that static pictures can't. So you've got to do it, all right? I think that's it. You may now do your, make sure that you get your Pearson's online assignments done that are associated with Chapter 16. All right, we'll talk to you later. Bye. Let's see if I can escape here. And that's it.